Hi, everybody. My name is Chris Bowser, but everybody just calls me Bowser. That's fine. And I have kind of an interesting role with Cornell. Yes, I am a Cornell Water Resource Institute staffer, but I'm closely partnered and within the New York State Department of Environmental Conservation, particularly something called the Hudson River Estuary Program. That's my email right there, but you can also reach me through Cornell or anything like that. Um, and my job is wonderful because I get to be both an educator and a scientist. Science communication is what I love to do, but not just because I like to communicate about science, but because I love to sponge it in. One of the things I've noticed is that sometimes it's good to take a minute and kind of like figure out where we are in the scope of today's project. So before we even get into Matt or Dr. Hare's work, let's just think a second where we all are, where you are. You may not even be on this map. Many of you are in Ithaca. I'm at the Norrie Point Environmental Center. And I know that at least a couple of you are in New York City, and I have a funny feeling that our presentation will lead us down to the city. So we're all pretty familiar with the watershed concept. And the Hudson River watershed is very, very large. And what I want you to think about is when Matt starts to talk about things like turbidity and things that are in the water, remember that this entire watershed is basically a valley that is going to bring us to where the setting of today's presentation is. Some of you are also familiar with the term estuary. The estuary is that part of the Hudson River that is an arm of the sea. It is directly connected to the ocean, basically from the Federal Dam at Troy, 155 miles south to New York Harbor and the Atlantic Ocean. The Hudson River ebbs and flows with the tides. In fact, the Stockbridge-Munsee Mohicans and the Lenape-Munsee of this area, they have several names for the Hudson River. One of my favorites is Mohicanituck, which some people translate as the river that flows both ways. If you get really deep into it, it actually means the waters that are never still. And I love that recognition of the concept of the ebb and flow of the estuary. Because you have this ebb and flow between ocean and watershed, salt and fresh water, you also can't talk about the estuary without talking about salinity. That salt that is coming up the Hudson, but getting diluted as it goes up. And that also helps us to focus a little bit on our talk today, because we're going to be talking primarily about salt and brackish water. And I'm sure that Matt will be getting into that. But we're sort of, our watershed concept is coming down, down, down. We're getting into much more of a concentrated area into New York, New Jersey Harbor, the Raritan Estuary, the lower Hudson River, places that have phenomenal historical context going back literally 10,000 years or more. Why is this place so important? We're going to find out, and we're going to find out about some of the organisms that live there. Now, my own journey with oysters has sort of gone all over the place. One of the tremendous projects that I get to manage as part of the DEC and Cornell WRI is something called A Day in the Life of the Hudson and Harbor. There's a couple people on this call that know exactly what I'm talking about because they are in this event. Every year, usually in early to mid-October, 5,000 students and teachers converge on 90 sites along the Hudson and Harbor to study the river on one day with nets, with water quality meters, with sketchbooks, most importantly, with minds and hands and hearts. What's neat about this project is that it involves a lot of partners that are all working on their own individual specialties. And one day I was visiting some of our Day in the Life of the Hudson and Harbor partners, and I found myself at the Hudson River Park Trust on the west side of Manhattan, where they have this beautiful oyster tank. And they were using live oysters to basically demonstrate the filtration power of the oyster. And what was really so relevant about this is that the oysters are, as we're going to find out, just sort of like signature, keystone, flagship species of New York Harbor. So overhead is this beautiful architecture of these scintillating skyscrapers, and underwater potentially is a different type of architecture, a living architecture with these tremendous ecosystem services of filtering New York City's water. In fact, it kind of made some big news a few years ago when the Hudson River Park Trust found this enormous croc-sized, and I do mean the footwear croc, oyster. It's I can't remember the name of it, but I can look it up and find that out. As also part of day in the life, I sometimes find myself over at Governor's Island. And Governor's Island is home to a couple of great organizations, including the New York Harbor School, therapy, and a tight partnership with something called the Billion Oyster Project. And the Billion Oyster Project is devoted to restoring oysters to New York Harbor for their purposes of shoreline stabilization, their purposes of filtration, and their purposes of habitat. And if you ever get a chance to visit the students, the high school students and teachers that are putting this together, you'll realize we are literally looking at professional scientists, professional engineers being crafted as we speak, as they check these oyster cages for the different animals that live there, for the different metrics that they can study. So it was really fascinating for me to kind of learn about oysters through the point of view of students teaching me. And as an educator, I can tell you something. The best feeling in the world is when you as a teacher can be quiet and the so-called students are themselves the teachers. And one of the main lessons that these students brought to me was the incredible importance of oysters as habitat. All of the different organisms and creatures that are thriving in these oyster beds. And it has really given me a huge appreciation for this very, very humble animal. I mean, honestly, what is more humble looking than an oyster? It looks like a flat rock. It feels like a flat rock. And yet, when you start to dive deep into this topic, it is mind-blowing, which is why I am so honored today to introduce our featured speaker, Dr. Matt Hare of Cornell University. You can look up his full resume online, and he has done some amazing, amazing work. But there was one sentence in particular that I wanted to call out because it really spoke to me. And it has to do with Matt's work as the curriculum chair of environmental biology and applied ecology. As director, Matt, Dr. Hare, promotes curricular development that maintains the unique blend of a balanced interdisciplinary core, concentrations that promote deep training or innovative bridging, and experiences such as internships and a capstone course that prepares students for integrative work experiences. The standout word, and it comes up at least twice, maybe 3 times, is this idea of integration. And I think that is the perfect bridge to talking about oysters, because if there's anything that integrates land to water, that integrates healthier waters, that integrates habitats together, it's oysters. So I am really looking forward to, just like the rest of you, to having a great presentation with Matt, a presentation that I believe is titled Oyster Population on the Hudson, Critical Knowledge for Restoration Planning. Folks on the call, put your digital hands together for Dr. Matt Hare of Cornell University.
Thank you very much. That's a wonderful introduction, and I love your emphasis on integration at the end there, because that's what I'm going to be doing in this talk, is pulling together a lot of different threads. I want to, on my title slide here, point out that I'm not working alone on this. The Hare Lab includes this recently, Henry Hua is a senior now in the environment and sustainability major that Chris mentioned there. and Harmony Borkart-Greer is my manager, my lab manager. Sean Kramer is the mathematician that's been working on the biophysical models that I'll be describing and showing you. So it was about 50 years ago that the Federal Clean Water Act was passed, initiating many of the regulatory and infrastructure improvements that have led to positive water quality trends like you see here for dissolved oxygen. And so that enables the possibility of restoring more ecological function to the estuary. But it's not just about the ecology in the water. Social ecological vitality has increased as a result of these water quality improvements, such as in the Hudson River Park, where they've been restoring old and derelict piers to create recreational green space in places where it would have been unthinkable a few decades ago when the Hudson was pretty reliably smelly. So there's been this visitation that they get in the park now that they've created this beautiful human habitat. They measure that visitation in the millions per year. And they also have a big focus on outreach and education. This is Hudson River Park Trust, which helps build an appreciation in New Yorkers for their aquatic surroundings. And with that appreciation comes a constituency willing to vote for and fight for city policies and infrastructure that can nurture the surrounding environment rather than polluting it. So I'll describe the partnership that I have with Hudson River Park in my research. Billy and Oyster Project is another partner. They've been supplying me with shell for our monitoring work, and they get that shell by the hard work of going around to all the restaurants and recycling it, collecting the shell once the folks have eaten the flesh out of it. And I really like the founding principle that Murray Fisher and Pete Malinowski had when they started the Billy and Oyster Project, and that is the idea that restoration without education is only temporary. I'm very focused as a scientist on sustainable aquaculture and sustainable restoration. That is, I want to see populations that are self-sustaining, that we don't have to keep planting oysters from hatcheries. And I totally agree with the Billy and Oyster Project that also requires that our coastal population of humans is engaged and willing to do what it takes to maintain those healthy environments and healthy populations. So why oysters, though? Well, oysters, as a function of their filter feeding, they cycle a lot of nitrogen and phosphorus and sequester, meaning they kind of pull it out of the water column and lace the bottom sediment with this nitrogen and phosphorus. Coastal waters are sort of over fertilized with these chemicals. So this is a helpful way of improving water quality. As oysters build up a reef structure that provides habitat for hundreds of other species, including some commercially important fishery species that use estuaries as nursery habitat. And these 3D structures can also attenuate storm surge. Just the fact that they're filter feeding microalgae out of the water allows sunlight to enter the water deeper, which some other organisms like seagrass really need. So for all these reasons, oysters are sort of this keystone species where if we can restore oyster populations, it's going to synergize and help with the vitality of the entire estuary. Chris, thanks very much for defining estuary for me. So as I talk about the estuary, I'm really talking about that zone that's described in this map. Although I'm not showing a little bit to the south of Manhattan is the ocean, and oceanic salinities are about 30 parts per thousand salt, and up by this yellow portion near Tarrytown, a little bit above Tarrytown. The proportion of salt is getting down below 5 or just 1 or 2 parts per thousand. And so this is the zone, the brackish zone, where oysters like to live. So it used to be that this Hudson River Estuary in the early colony days was just the coast was just inundated with oyster populations, and they were a food source for people in New York, and they were known the world over as a source of an amazing culinary experience. Currently, this yellow tappan zee population that I depict on the map is the only self-sustaining wild remnant population. And in addition to that, though, there are populations that are being established by the Hudson River Park and by the Billy and Oyster Project, where either in the areas of the river that are recruitment limited, meaning there's not very many baby oysters that can populate new habitat, then they use hatchery facilities and aquaculture strains of oyster to, it's called seeding materials like bags of shell to sort of get a population started. Whereas up by the tappan zee population, there's a lot of recruitment, a lot of breeding and young oysters, and in that area, a lot of these hard habitats were put down onto the bottom of the river that quickly became populated with oysters, with the local wild oysters. So with that background, our research questions in the Hare Lab are motivated by a desire to protect this tappan zee population into the future when it's projected that climate change with an increase in both the frequency and magnitude of storms may make that marginal low salinity habitat less ideal for oysters. They're already kind of at the edge of what this species, eastern oysters, typically like. So specifically, we're interested in why is this remnant tappan zee population remaining isolated up in that marginal habitat, even while water quality south of that has been improving? How can we leverage the recruitment that is each year's crop or cohort of juvenile oysters? How can we leverage those from the tappan zee population to expand the population southward? And what sites southward should be prioritized for restoration to develop a highly connected metapopulation? So I want to start by giving you some life history for the eastern oysters, because that'll help understand what I'm meaning by connectivity and why it's important. And then I'm going to show you the patterns of juvenile oyster settlement that we have found since 2018 that have generated a couple of hypotheses for why the tappan zee population might be isolated. I'll then introduce a biophysical model that is allowing us to make predictions about the spatial settlement patterns of each new cohort of oysters, where they're ending up, where we expect them to end up along in the estuary. And then I'll be showing you how a new genomic tool can help us actually map out the connectivity patterns between different patches of oyster habitat in the estuary. So oysters, even though they do look like a flat rock, these are amazing organisms. A single adult female, not even as big as the oyster that Bowser showed you, but just a 3 or 5 year old female will pump out millions of eggs each summer. So very high fecundity. All the oysters in an area sort of trigger each other for this spawning. So they're synchronous in spawning. They're all putting their gametes out in the water column, and that's where fertilization occurs. And this starts the development into larvae that swim around for 2 to 3 weeks eating microalgae. They're not strong swimmers, so they can't swim against the tide, but they can swim vertically. And estuaries tend to be vertically stratified. So there's a freshwater lens heading out towards the ocean, while the tide is bringing up a saltwater lens underneath it. And that vertical stratification means that just a little bit of vertical migration by a larva gives it a lot of capacity to move up or down in the estuary. So along with that high fecundity is very high mortality while they're in the water column, and then soon after they go down to the bottom, find suitable substrate and cement themselves down. And so then after that, after those first few weeks, when they metamorphose, they no longer are moving around, they're sessile. So since 2018, we've been planting these mesh bags of shell that my crew here is holding near the truck, collaborating with the Billy Oyster Project and a team called the BioBoat to deploy these throughout the lower Hudson River. And Billy Oyster Project was often working further south in the estuary in a complementary monitoring. And so these are like monthly deployments during the reproductive season of the oyster, July to September. And what I show in this map is 1 year of data that kind of shows the characteristic pattern that we tend to see most years. And that is the highest abundance of juveniles, and what you call a baby or a juvenile oyster is a spat. So we'll find hundreds to sometimes thousands of spat on a pair of shell bags up near Tarrytown and the Tappan Zee Bridge, and then quickly diminishing numbers as you move south into Yonkers and the upper Manhattan, and then often just a handful down in lower Manhattan. And so we've been interested in what's causing this pattern. So deployments are pretty boring. We're just putting out these dry bags of shell, but the recovery of those bags is very exciting. And that's when we like to engage community scientists to help us search through the shell and not only find the spat, the juvenile oysters, but lots of other critters too. So it's a wonderful educational learning opportunity. It's just a fun discovery opportunity to find, the crabs and the worms and tunicates that are in there. And so this is that same place in the river project of Hudson River Park, their wet lab. You can actually see the tank that Bowser showed you in the right-hand picture here. And this is one of the days in which we engaged community scientists to help us find, measure, and count the spat on the shell bags that were collected from throughout Hudson River Park. And with their help, what we found in 2022 is this pattern. So I'm showing you a graph where the histogram bar, the red bars are indicating the number of spat per shell. The tallest bar there means that on average we saw 4 spat per shell, and then there's maybe 200 shells per bag. And that's up north near the now the Mario Como Bridge. And then pretty quickly it drops right off and diminishes, so that in Hudson River Park we saw very, very few per shell bag. So that is the pattern we see most years. In 2023, though, things got changed up. Our where we were putting the bags changed a little bit because we were testing a hypothesis in Hudson River Park, and so we needed more deployments in the Hudson River Park. But the graph now for 2023 still spans from Tappan Zee, where you see a very small bar, almost zero. So we found very few spat up north, and an increasing number as we went south. The numbers that are along the chart, let's see, I can get a pointer here. There we go. So these numbers show a bit of a trend there. So these are indicating average height in millimeters for the spat that we're finding. So these are pretty small guys, about 3 to 4 millimeters up north and as large as 8 millimeters down south. So it's not clear what's going on there. If the whole river was synchronous in when they were spawning and producing larvae, then it would suggest that perhaps down south, spat are growing a little faster. So we're finding them after a month to be larger than up north. But it's possible that they're not entirely synchronous throughout the whole river. And so that there was slightly different timings for the spawnings that generated the spat that we're finding in different parts of the river. So these are the sorts of monitoring data we're getting. And I also want to explain that this really tall bar in Hudson River Park, what we were doing is testing a hypothesis that places in the park, like in the Intrepid battleship that are sitting on the bottom, might generate an obstruction to the tide that creates a lot of swirling of the water. And some previous data on from divers that found a high density of oysters right near the Intrepid made us think that that might generate greater numbers of settling oysters on average. And so we're testing for that near the Intrepid with these 4 sites and near Gavesoport Pier, Gensford Pier. And so in each case, we had 4 pairs of bags around those possible baffles, and then 3 of the light green control sites. Unfortunately, this one control site, most of the bags are hanging from a dock, and so they're a couple feet under the water surface. This tall one fell down to the bottom by mistake. It just and ended up having 4 times the amount of spat. So this is an idea where in science, there's often an element of serendipity where you learn things when things go wrong. And so this is not very helpful data in terms of a control for this experiment, but it's really helpful for us to realize that if we do our deployments down near the bottom, we may be able to capture a whole lot more spat for a given deployment than with our current methods. What we did end up finding is that there was no elevated settlement in 2023 near the Intrepid relative to the closest control, or at Gansevoort Pier relative to its closest control. So no evidence there to suggest that our hypothesized mechanism is at work. So we still have the large scale question of when oysters, these oyster larvae are in the water, why are they mostly staying up north? And the patterns of recruitment that we've seen suggest 2 hypotheses. One is that perhaps larvae are dispersing southward, but as they move from the Tappan Zee area down towards the city, they are going through a gauntlet of both treated wastewater and untreated wastewater, and a lot of other possible pollutants. And so it's possible that they're just not surviving in great numbers as they move south, or the physics of the river in combination with the behavior of the larvae may be keeping them up north. And so at least this second hypothesis is something that we can test by using a biophysical model. And what I mean by that is the physical part is using the New York Harbor Observing and Prediction System, NIHOPS. It's a very well-validated model that models the physics of the tide, the discharge from the Hudson River, from that whole watershed that Chris showed you, the effects of the wind, and basically everything that's pushing water around is in the model. And then in addition, because larvae do behave as I described, we're actually incorporating into the simulation tracers, in silico larvae, if you will, that behave as best we know, behave the way we think oyster larvae behave. So there's some technical specifications here about how the model works, and I'm not going to spend any time on that. But as I say, it's validated in the sense that if you put an observatory out there and measure temporal changes in salinity or temperature, the model recreates those pretty well, and that's all documented in the literature. But this oyster larval part of it has never been done before. So let me just show you with a cartoon what these larvae are doing in general. Whoops, let's see. So the larvae are formed down at the bottom where the broodstock are, and then they, as very early stage in the larval development, they swim up towards the surface and become kind of stabilized behaviorally at the region where there's a sharp halocline, if there is one. And so a halocline is this shifting in the salinity because, as I said, there's fresh water and relatively low salinity on the top and salty water coming in with the high tide on the bottom. All right, so that's where the oysters tend to sit for a while. They're eating a lot of algae there for 2 to 3 weeks. And then when they become competent, that is physiologically competent to find habitat down in the bottom and metamorphose, then they go down there and they search around and they glue themselves to something hard. So what we're doing with the model is arbitrarily separating out different polygons of either where tracers are being released or where we're keeping track of where they end up after 2 to 3 weeks. And we're releasing these tracers within the model between July and August, which is when our data suggests that there's oysters reproducing in this river. The releases are actually 5 different releases because the phenology, the timing of reproduction does tend to get, it's not just one punctuated synchronous spawn. There tends to be multiple spawns, with the biggest one being in mid-August. So that's the biggest release of these in silico tracers. And so I'm going to show you particular release locations up near the Tappan Zee region. These at the ends of the particular domain that we're focused on here are areas where I'm not keeping track of whether the tracer is settled on the east shore or the west shore. It's just kind of treating it as sink habitat. And so let's look at a situation where we're tracking tracers in the model for 1993 conditions that were released from these 3 polygons. And then the graph here depicts where along the north to south shoreline the oysters settled and ended up in the model. And so the the blue histogram on the left of this plot is settlement on the west shore. So New York and New Jersey. The red histogram on the right is the east shore, New York and Manhattan. And this bar up at the top, the gray bars at the far top and the far bottom, are these sort of sink habitats outside of the domain where we're looking at high resolution. All right, so this yellow depicts the release location. And what you can see here is that even though they've been sloshing back and forth with the tide for 2 to 3 weeks, they're all pretty much remaining up near the breeding population. We're not seeing settlement on the west shore or the east shore. When we only look at the breeding release, breeding activity from the western part of the Tappan Zee area, they stay up north. What if we look at releases from the east shore? Because there are oysters living up in the Tappan Zee region on both sides of the channel, of the central channel. Well, here we see much less, almost no oysters ending up on the western shore. Most of them end up on the eastern shore or far to the north. And but here again, very few of them head south and end up in areas below the Tappan Zee population. So what this implies is that it's the physics of the river that are maintaining this isolated population up in the Tappan Zee region. And if we want that population to spread to the south, we may need to do some manipulations and help them out, that the physics of the river in general, in most years, are not going to spread them around that much. That's pretty good to know. And actually, so I have not tested whether there's a pollution gradient that is causing any part of this effect. That's would be yet another study and still needs to be done, because that's also still possibly a complicating factor. So now coming down to the Hudson River Park again, And so now this is the lower west side of Manhattan. In 2021, they planted over 1 million seed oysters in a restoration that was established there, as well as several hundred of 2 other different kinds of aquaculture oyster. Those were put in as adults, whereas these seed oysters were put in as juveniles. So a juvenile oyster, a spat, needs a couple of years to become reproductive. And so that means that in 2023, these millions of oysters, however many survived, are ready to reproduce. And we were very interested in working with Hudson River Park to see whether we could determine from the model how many of those we're expecting their larvae to actually come back home and resettle in the park from restoration oysters in the park. And so we did that again by using the model, but now the release of in silico tracers is only from this region down in the southwestern part of Manhattan. And I wanted to show you how the 5 different releases in any particular year, and this year is 1993, the 5 different releases tend to be at different parts of this tidal cycle, and that generates quite a bit of variation in how much of the settlement after 2 to 3 weeks from this one release location is on the west shore or the east shore, and also how much of it is further north, like this is up near Irvington, versus to the south. And so this is the blue, the gray bar here is showing how many oysters got flushed to the south in this checkered area. So there's quite a lot of variation there, which we account for by just averaging it all to show you a single year pattern. And here we were interested in comparing 1993, a relatively low discharge year for the Hudson. There just hadn't been much snow that year, versus medium versus high discharge year because of storms and snow melt. And what you can see is when the release is down in Hudson River Park, most of the larvae are moving north and settlement is happening along Manhattan and up around Irvington. So they're heading up back towards the Tappan Zee population, mostly settling on the west shore, but particularly when there's low discharge from the Hudson River, there's some landing on the east shore as well. And you can see that with a high discharge year, there are a greater number of oysters getting flushed down south of Manhattan, and we are now expanding these studies to understand exactly where these are going when they go south of Manhattan. So I want to emphasize that these are very simple models. Okay, there's a lot about larval behavior that we don't know. There's a lot of features about this recruitment process that we're not modeling. So we're not modeling larval mortality, larval sensitivity to low salinity or to pollution. So all we're doing is studying the potential for movement and spatial patterns of settlement. But that still can be really valuable in the ways that I've shown. Now we have a reasonable understanding that the physics of the river could explain the isolation of the Tappan Zee population. And we have to deliver this unfortunate news to the Hudson River Park that, at least according to the model, it might be that they're in this little spatial window where there's not a lot of larvae that are going to come home there. So they can be a source of larvae to the rest of the river, and particularly to the north, but they shouldn't expect, based upon the model, a whole lot of larvae coming back. Now, I talked about this model and the physics being validated. I feel like it's pretty important to validate this, this biophysical result and this prediction about larvae. And so what we've done is moved to genomic tool in order to do that. And the principle on which the genomic tool was built is the idea that when you take wild oysters and bring them into the hatchery and start a breeding line, which is important for aquaculture to breed them for commercially valuable traits like disease resistance and fast growth. Every time you do that, you're putting millions of larvae into these tanks and culturing them in a very different environment than what they're used to. That inevitably generates very strong selection for traits that we're not necessarily measuring, as well as the traits that are ultimately put under selection for the commercial value. All right, so this is part of what we might call domestication, very early domestication. And in work in my lab, compared at the whole genome level, genetic variance in wild oysters relative to selected strain aquaculture oysters, and found that found over 1,000 genetic variants that were significantly different in the allele frequency, in the frequency of the variant in these two groups, wild versus selected strain aquaculture oysters. So what we're looking at here is it's called a Manhattan plot because it often looks like a skyline. There's 10 chromosomes in the eastern oyster genome, and so those are along the x-axis, and on the y-axis is a measure of the statistical significance for basically the amount of allele frequency difference between wild and selected strain oysters. And so the red dots are these 1,100 highly differentiated variants, differentiated between wild and selected strain. And the very thin vertical lines are a set of 37 of these variants that, using machine learning methods, seem to be the most efficient way of easily distinguishing these two kinds of oysters. They don't look different, and genomically they're very, very similar. But whatever's changing in this early domestication process seems to be captured by, at a minimum, these 37 different variants. So we use these 37 variants to characterize a bunch of wild population samples from North Carolina to Maine, and a bunch of selected strains that different groups have generated over the years, and that are being used in aquaculture. And you can see in this discriminant function plot, it's basically a multi-dimensional analysis. We've got 37 dimensions here because we've got 37 variants that get scored in every one of these oysters. Every dot here is an oyster, and every ellipse is characterizing the spread of the dots for a given selected strain or a given wild population sample. And what I want you to see here is that the red ellipses and the blue ellipses are mostly distinguished in this bivariate plot when we look in that tries over 2 of the 37 axes in multidimensional space to characterize the variance in this genetic variation. And so what it's showing us is that we can distinguish with these 37 variants these two different populations of oysters. All right, so why do we care about that? Well, we care because we're studying a river system where we've got this remnant wild population. So that would look like these blue oysters. And where there's restoration in the Hudson River Estuary, it's using these aquaculture strains. So right now we basically have 2 point sources in the lower Hudson River, where from the north there's wild oysters coming into the system, and from Hudson River Park and other restoration sites in the estuary, there's the possibility of getting juvenile oysters that come from a selected strain. So we basically, with this genomic tool, we can separate these two kinds, and for any one juvenile oyster, figure out probabilistically which population it was more likely to have come from, and therefore which area in the river it was most likely to come from. All right, so this is a plot where I'm just showing you for the Tappan Zee wild oysters on the right, and the two of the 3 strains that were planted in the Hudson River Park. It's the same sort of plot in which we're looking at dots being individual oysters, and the spread of green dots, for example, here for this one aquaculture strain is the spread of the genetic variance on two axes of the multidimensional space that describes all that genetic variance. And the important part here is that I'm showing you that a new tool that we generated from these 1,100 informative variants to make for an efficient genetic assay does a decent job of separating these two types of oysters. So now if we use this to analyze the spat that we found and analyzed from 2022 around the river, we see those 2022 Hudson River Park spat lining up on this plot and mostly obscuring the blue wild reference samples. None of these Hudson River Park 2022 spat ended up over in this part of the plot, which would have suggested that they have aquaculture parents and would have come from the restoration site. Instead, they seem to have all come from the northern breeding population, the Tappan Zee population, and settled in Hudson River Park where we collected them. Okay, so that's 2022. I told you that the bulk of the oysters that were planted in Hudson River Park were juveniles in 2021, and so they're going to start breeding in maybe 2023. So now let's look at a plot with those 2023 oysters. You might expect maybe there'll be some that show a aquaculture signature indicating that they're spat collected in 2023 that had parents in Hudson River Park. But in fact, we see the same thing. We see that most of them are coming from the Tappan Zee population because they have this genetic signature that is indicative of wild oysters as opposed to aquaculture oysters. Now, there is some of intermediates where there's a little less certainty. And so at this time, I can only say that the broad preponderance of spat are showing this wild signature. And therefore, the genomic data are reinforcing what the biophysical model predicted, and that is not a whole lot of baby oysters coming from the restoration oysters in Hudson River Park are actually coming home back into the park. Instead, the park is getting most of its recruitment from the Tappan Zee population up north. All right, so what does all this mean? So I showed you that oyster recruitment is concentrated up north near the Tappan Zee breeding population, at least in most years. There's some odd years where there's a different pattern, and we're still trying to understand what's generating that odd pattern. The biophysical model indicates that that the physics of the river can explain this high recruitment, high self-recruitment up at the Tappan Zee population. And it also predicted that there is relatively low self-recruitment for restoration oysters in Hudson River Park. And I then showed you that genomic analysis has the power to distinguish these two types of oysters and give us the spatial resolution to say that in both 2022 and in 2023, most of the spat that were found in Hudson River Park had parents up in the wild Tappan Zee population. So our continuing analyses will focus on the third aquaculture strain that is the most abundant strain that is in the Hudson River Park, and determine whether any of the 2023 spat perhaps have Muscungus aquaculture signatures, and indicating that they're so that we refine our conclusions to indicate that there are some restoration oysters coming home to Hudson River Park. And so far these results are tentative. But the implications are that the pattern of oyster recruitment from the Tappan Zee population is mostly that they're isolated and they stay up in the north. Only sporadically and occasionally do they come far to the south, that is like southern Manhattan. And so it's important, I think, to identify these forcing factors. My hypothesis is that when there's storms that occur while the larvae are in the water, so in the July-August period, that may influence the spatial distribution of where the larvae end up and oyster settlement happens. So we'll be using biophysical modeling with data from 2022 and 2023 so that we can make a direct comparison to our spat observations in those years. So in most years, oyster larvae settlement in the South seems to be mostly from the northern Tappan Zee population, but we need to expand our genomic testing to other areas of the estuary where we might be seeing more of a contribution from the multiple restoration reefs that the Billion Oyster Project have been working on. And finally, that's our goal, is to map the areas that have the greatest potential for larval exchange and settlement, because these would be a high priority for spatially siting areas where habitat should be restored, and in order to invite and promote oyster recruitment and a fully a meta population, that is a population of reefs that have a maximum amount of connectivity among them, given this life history. So I want to thank the many agencies that have provided funding, including the Water Resources Institute and the folks that have helped in the field, as well as our collaborators in the Billion Oyster Project and Hudson River Park Trust. And thank you very much for your attention. I'm